And so like if you miss a bunch of points on exam, your homework grade and your lab participation is going to bring up that exam grade. I think looking globally across the class, everybody in class has an A, a B, or a C kind of as your running total right now. We've done about a third of the material. We've done about 30 points worth of material. It makes you feel better. I had a student that like honestly failed this class last semester. Like I have almost framed her final exam because I was just like, wow, how could we have so miscommunicated about this information that you would write that on the exam? So none of you are in that range at all. You're not giving me answers that are so incorrect that I want to enshrine them. It makes you feel better.
Or number 33, it had like the graph and the climb.
Yeah.
Like, what was the correct answer to that?
It was A.
A lower production.
Lower nitrogen means lower photosynthetic rate, which means it will grow slower over time.
That makes total sense too.
It's a question that the graph has almost nothing to do with the answer. Okay. Are you guys ready to move on? I certainly consider the first portion of class, it's about environments and abiotic conditions, and it's about evolution and how species change over time, and thinking about conditions and environments as speciating factors, as things that influence species. What we're going to talk about for the rest of this semester is what I consider kind of classic ecology. And so we're going to be population and community focused. Recognize that populations are subsets of species. So we have to understand what a species is and what influences species, which is what we have done. And then communities are species put together. So again, this like species and individual is the basis of what's happening in ecology. But we're going to turn to kind of hardcore ecology now. A lot of the stuff that we're going to talk about in the next few classes, we have already encountered in lab. And that's because we need to get out in the environment and do lab when the weather is 90 degrees and there are leaves on the trees, rather than in November when you can't tell what plant is which because they all look like dead sticks. Okay. So there's been a bit of a mismatch between what we've been doing in lab and what we've been talking about in class, and we're going to catch class up a bit of what we've been talking about. Okay, we're ready to talk about populations. We are in chapter 8 of our textbook, and we're going to go chapter like 8, 9, 10 as we move through the next few weeks of class and as we go through fall reading days, which I don't know about you, I'm very excited about fall reading days next week. We get 2 days off. Okay, but our first purpose that we need to do is talk about what is a population before we can grow and change it and manipulate it over time. Okay, all right. So populations are made up of organisms, and our textbook goes to great lengths to point out that there are different ways to think about organisms, and this is going to play into then how populations are distributed in environments and how populations grow and change over time. So thinking a little bit about organisms and individuals and life histories of organisms, where we'll eventually end up with this, helps us understand some of these things that we're going to start to observe and learn about populations. So an organism, of course, and we think about an individual organism as a genetically unique individual. And hopefully in biology, it was stressed to you that in sexually reproducing organisms, sex is the great recombinder and the great producer of unique individuals and genetic variation. You have crossover between chromosomes. You have independent assortment of chromosomes. You have unique fertilization between different gametes from different parents. And in this type of unitary individual for organisms, we have vertical transmission of genes from one generation to the next. And each individual is a unit of that population. They're genetically distinct from all the other individuals in that population around them. Does that make sense?
What do you mean by vertical?
So that's like parent to offspring, okay? You don't have any horizontal inheritance across generations, which is what I'm going to talk about next, okay? All right, so like this is normal. And we're like, yeah, that makes sense. How would it be any other way? Because this is what we do, okay? But There are lots of organisms that do this a little bit differently, and as a result, their populations are going to grow differently and respond to environments differently than if you are a unitary individual within your population. Okay, all right. In modular organisms, okay, they grow individually and then as a population differently than unitary individuals. In modular organisms, they have horizontal growth and inheritance. They transfer genes directly to their sibling, and that's usually because their sibling is a genetically identical clone to them, okay? They're not moving stuff one generation to another generation. That happens too, but one of their main growth processes is to bud off individuals that are genetically identical to them, or to send out a shoot, either underground or above ground, that grows a genetically identical individual, whether it's a plant or an animal. This kind of modular reproduction mechanism is what happens in all of our single-celled organisms. When they go through binary cell division, they produce a genetically identical clone to themselves. Okay. Oftentimes in modular organisms, they have multiple pieces of their body that can act as individuals, but they're all connected and they're all genetically identical. So in the little coral head, we have all these little polyps of coral that come up. That's all one organism. It's all genetically identical little coral polyps that have budded off from their neighbor. Okay? We can think of the polyp as an individual, but really we need to think of the coral head as an individual, because that's what's genetically unique from other individuals in the population. The same thing with our sponge here. It has a modular body. Each of these we might, harvest and sell as a sponge. Okay, they're each feeding independently of each other. They're each going to make gametes independently of each other, but they all came from one sponge that sat down and then has grown new body parts to the side. Okay. The same thing here with the strawberry plant. It's sending out shoots that allow it to start new little strawberry plants to the side. Okay, that strawberry plant is an individual. It can grow and reproduce on its own, but it's genetically identical to the starter plant. And so if we want to talk about genetically different individuals, this whole thing is one individual, even though it's got different little body parts. It's a modular individual. Okay, does that make sense? Okay, cool. We can talk about genets then in modularly growing organisms, genetically different individuals. Strawberry plant over here, 50 feet away, different strawberry plant that's not connected. One coral head versus another coral head, those are genets. The ramets then are the clonal organisms, the different modular parts of the body of the genet. Okay. So ramets are genetically identical clones. They're all the little polyps on the coral head. This affects then how populations grow and change over time. If you're capable of modular growth, you might be able to expand your population really quickly because you don't have to go through the sexual reproductive cycle to do that. It also means when we're thinking about genetic diversity, we can grow lots of coral and repopulate a reef, but if they're all genetically identical to each other, we've only changed species abundance. We haven't really affected genetic diversity in a way that we probably would want to. And so depending upon your organism's life history, this type of idea becomes important, both for understanding organisms and making predictions and for management types of issues of populations.
So are humans, does that term apply for us, genets then?
Yes, we are all genets. Okay. So in an individual unitary population, everybody's a genet. Okay. The only place where you would have a ramet potentially in humans is if you had identical twins. And then they would be ramets of each other. Okay. But in reference to everybody else, they're genets.
Gotcha.
And we don't ever talk about that way, but you could.
Okay.
All right, a population then is all the individuals of the same species that live in a given area. That's a population. And when we do conservation biology, we are often very interested in population. A population shares a gene pool. There is gene flow between all the individuals of a population. And that is usually what helps us to define the boundaries of a population. When gene flow breaks down or becomes lessened, then we say we've moved into a new population, even though it might be the same species. Okay. The other thing that really influences what is a population is the geographic area. So the population of this room is those of us in the room, but we are members of a larger population that includes everybody on campus, and then everybody in the town of Batavia, and then the greater Cincinnati area. Your scale of spatial definition of geographic area will influence population size, distribution, density, how that plays out. And I'll give you a couple examples right here. We can describe populations then in several different ways. And so in our field studies out of the Cincinnati Nature Center, we've been looking at populations, and we've been really interested in population abundance, right? How many of this plant is in this area? How many of this particular type of tree is here? That plays into distribution, and the abundance and density of a population will be a factor of not only population size, abundance, but also how that organism is distributed across an area. And that varies with life history and species to species. Population growth rates, population distributions can really be a factor of age structure, as we're going to learn to talk about in the next couple weeks. And understanding populations and age structure and distribution and population abundance now helps us predict population growth. And so today we're going to look at some different abundance ideas and density distribution patterns to get us set then to be able to talk about things like age structure and growth. One of the really basic foundations of ecology then is assessing population abundance. Okay, so if you had these this pod of dolphins here, and it's probably on the superpod scale in terms of cetacean ecology, how would you count all the dolphins? How would you get an idea of population abundance?
Tagging, maybe a long time though.
Thank you, tag, and we'll talk about like how that we're going to talk in detail about tagging and how that works today. How else could you do it?
If you take like a sample population and then expand it.
Right. That's the basis of all of our population assessments, is that we count the individuals in one area, and then we multiply that by the total area of their geographic range. And we say, hey, there were 10 here. There's 10 kilometers out there. I bet you there's 100 individuals in the population. That works, but there's always a caveat, okay? And so like you could take this picture of the dolphins and you could count all the dolphins you see, and you'd be like, okay, there's this number of dolphins in the pod. The pod was this big. I had a bigger picture of the area. Dolphins spend a lot of time underwater. Okay, so you're missing all the individuals that are underwater at this instant in time. And so in cetacean biology, we usually count the number of individuals that are on the surface, and then we multiply it by a factor of 3 for all the individuals that were underwater at the time that we counted at the surface. And that you have to understand your organism to be able to do that multiplication factor, to know what the right factor is, okay? To know like how many you need to count to get a good count of population. Okay, all right. So populations are distributed across their particular geographic range or their particular ecosystem. And when we're talking about population distribution, which is going to play into our calculation of population size or abundance, we have to consider 2 things. And they work together, but they are slightly different factors. And every once in a while, you'll run into an instance where you need to be very clear about the difference between these 2. The spatial location of a population is the area where it specifically lives. Okay. For this particular population, the area outlined in blue is its specific location. Okay, it lives in that little area outlined in blue. Okay. For this population, that spatial location is equal to its geographic range, an area that encompasses all the individuals of that population. Okay. The geographic range then is something that can be played with on spatial scales. Okay. And that the spatial location is where the organisms actually are at that moment in time, or where they're going to move throughout their life history. The geographic range is kind of like all the places they could be. Okay. All the places where the climate or the vegetation is right for them to exist. They might not actually be there, but they could be there. That's the geographic range. Okay. The habitat the physical environment and the biotic interactions determine the geographic range that's possible for any given species. And it's the habitat that physical environment and biotic interactions is often determining for the actual spatial location of individuals in that population. And this is where, again, the abiotic physical conditions start the process of determining what's going to live there. Okay. The habitat, again, just to stress one more time, it's both abiotic and biotic. It is both the non-living stuff and the living things. And we'll talk as we consider communities that we can set up a certain range of conditions where a particular species might live, and that's its habitat possibility, but then it often only lives in a fraction of that habitat space as a result of biology. And that's where competition and predation become so important to determining population size and abundance and distribution, because biology layers onto the physical environment. Keep in mind too that in considering habitat, it's not just what's necessary for the organism to live itself. We're also considering conditions that are necessary for it to grow and mature and then reproduce itself. And again, that becomes really important when we consider population changes over time. It's not just maintenance, it's also growth and reproduction. If we look at population distribution over space or over a geographic region, we can start to identify patterns in ways that populations and species tend to organize themselves across landscapes. And there are three general population distribution patterns that we see. And they are random, uniform, and clumped, okay? We're gonna talk about them like, hey, you gotta fit in one category. Your population is either randomly or uniformly or clumped distributed. The organization of individuals in a population, clumped, uniform, random can vary with life history stage, and it can vary on the spatial scale that you look. And so it's very much the snapshot and the specific conditions that you are talking about as to which of these categories of population is some, or if they are some mix of several of them. Okay. Definitely with random, uniform, and clumped population distribution, we're concerned about the density of that population, the number of individuals per unit area. So again, if you have a large population in a small area, you're going to have a very high density of individuals. If you have a very small population in a big area, you'll have a low density of individuals. And those two things, high and low density, sometimes affect population growth rate and things like competition and predation rates. And so density is just as important to know for your population as overall population size. But to know population and density, you've got to first have an idea of population size, the number of individuals actually there. Okay. And so in this idea, the spatial position of individuals relative to each other matters. Okay. If they're all packed in together, predation rates and competition tend to be higher, and there are consequences for growth and survival and reproduction. If individuals are widely distributed, that has consequences for competition and predation that might be positive, but it makes finding a mate harder. And it might actually then have a similar effect as high density when it comes to survival and reproduction. Okay, so in a random population distribution, like our field of wildflowers at the top there, the positions of individuals in the population are independent of each other. Okay, they are much more determined by something that's happening in the physical environment, like there's a patch of sunlight, so that's where the seeds can actually grow. It's not dependent upon the fact that there is a similar organism living right next to me or very far away from me. The classic example of this are the seeds that grow from a dandelion head when the wind blows away the seeds. Wherever a seed lands at good conditions, it will grow. Okay, it's random where those seeds end up. Organisms that are zooplankton and phytoplankton tend to have random distributions. They're being moved by the dominant wave and wind currents. They don't have any real control over where they're going to end up. Okay, the second category here is a uniform population distribution. And in a uniform population distribution, The organisms and individuals in the population are very evenly spaced away from each other. Okay. And this is usually due to negative interactions between members of the same species. So competition for space often leads to a uniform distribution of individuals in a population. You defend your territory, and then there's the next territory. You need a certain territory size, and that sets the space between individuals. Okay. Predation and territoriality, of course, play directly into this. And again, you may need to be a certain distance from your neighbor in order to reduce predation. And so you will be that distance apart. The classic examples of this uniform distribution are like in part B here. We're looking at a kind of overlook image of seabirds in a huge nesting colony. And each little nest is just the same little space away from each other so that it's the distance of two adults can reach to peck at each other. Okay, so like the bigger the bird, the bigger the distance between nests because they can reach farther to peck at their neighbor and try to shoo them out of their territory. The smaller the bird, the closer the nest because they have a lesser reach to peck at each other. Creosote bushes in the kind of southwestern United States is another example of this. Creosote bushes put out a toxin in their root zone that kills off other creosote bushes. And so whatever the maximum size that they can influence with their roots determines the distribution of the next creosote bush. And they have like a maximum size they can get to, and so you'll get bushes that are just really evenly, perfectly planted across the landscape, but they're not. This is due to a competitive negative interaction within the same species. All right, the final one here are clumped. And in clumped populations, the density of individuals occurs in groups. And you'll see many individuals with high density in certain pockets of their overall geographic range, because most often they're clumped around necessary resources. Okay, this is classic waterhole clumping of organisms on the grasslands of Africa. All the organisms clump around the waterhole because they must have access to water. They are much more densely distributed in the region near the waterhole than over the whole possible place that they could actually be in. The other thing that tends to cause clumping is modular growth of ramettes. If you're budding off genetically identical clones, that all happens in the space near you, okay? And that creates clumping pockets of high density. The example here, of course, are these reef fish, and they are clumped into schools of fish, both to avoid predation, and then also because they're clumped on the reef because the reef provides them habitat as well as food resource. So they're going to exist in a very clumped school formation. What kind of population distribution pattern do humans show?
Clump,
Why do you say clumped?
because cities that have resources.
Right. In today's modern civilization, human population is increasingly clumped into urban environments. This is the first time in human population, like these 5 years forward and back of us, when the majority of human population, just over 50%, lives in an urban landscape. And so that's clumping of 50% of the population into urban environments. That means that what happens in individual urban environments is really important to the human population because most of us live there, even if it's just like a tiny fraction of most to make it most, right? By 2050, 75% of us are going to live in an urban environment. And so managing our urban landscape is really important to human health on a like base level. What about in the past? Have humans always had a clumped distribution?
They said, travel west and take whatever land you want. Depends how far back we're going.
Right, exactly. And again, I want you to get this idea that this distribution has different scales that you can think about, both spatially, which we'll talk about next, but then also over time within a given species. And so if you want to talk about our, geographic range of humans, as we expand, we get more random or maybe uniform as we start to parcel out little bits of land that we can live in. And then as society changes, we tend to end up more clumped. But think that each individual family unit is a clumping of individuals. and that it usually, we tend to clump together in order to meet those basic needs of survival and to raise kids. I mean, I wish it took a whole village. I would like a whole village that helped me. But it takes a number of adults to get human children to adulthood, and that requires clumpiness. So again, the scale kind of matters. If you want to look at how we're distributed across the face of the planet, it looks quite random in terms of humans. There are some here, there are some there, but if you look, we're clumped around resources. The reason cities are where they are is because they had base resources that humans needed: access to fresh water, access to trade, access to agricultural land. That's why the cities are where they are today, because they were clumped around resources. Cool. Okay. All right. So this idea of interacting spatial scales in terms of population distribution, for some reason, I talked about the same stuff last class. I don't know why you guys got aligned to kind of the same topics at the same time. They got like really stuck on population distribution, and they were just like, oh my God, it matters. Like the scale you look at, like we're clumped in a family, but then it looks uniform if we look at the individual houses in a neighborhood,
Yeah.
but then all the neighborhoods are clumped again. And they were just like that's cool, right? And then they just wanted, wanted to talk about it. And I was like, guys, we gotta go on. But that's what this is showing you, this idea of interacting spatial scales, and you have to pick one, which is what my class did not want to do last period. They didn't want to pick one to just stick with. You gotta pick one. If you're going to study a population, you got to pick one of these spatial scales and stick with it. You can't flip-flop back and forth, or it's going to change how you view that population. All right, so you got this little moss. It's kind of global distributions. It can grow anywhere in these kind of latitudes that we associate with upper United States and Canada, as well as all the way across into Europe and Asia. If we look at the continental scale, though, we see that they tend to be clumped up here in the northeast United States, this little mossy plant. If we look then at this northeastern United States, we see that they have a pretty good kind of random uniform distribution, but then there's this clump right here. And if we look at that region, then we see that they're not randomly distributed, they're actually along rivers. Okay. If we look at an individual river, we see that they occur in kind of specific places along a particular river system. And then if we say, why are they there? It turns out that this has to be in a swampy wetland area on the edge of a pine barren, a place where you have pine trees that are either dead or in the process of dying. And it turns out that only on one side of the dying conifer stump will you actually grow this type of moss. Okay. And so your spatial scale of investigation matters in terms of how you're going to view and manage this population. If you're like, oh, this population can live anywhere, that's true. But if you want to really manage this population, you have to realize that it grows on one side of a conifer stump. Okay. And that's only where you're going to find it and where you're going to have to protect it. Okay. The other thing to keep in mind with populations is that we often talk in management and ecology about subpopulations. That yes, there is a whole population for a given species on Earth or in a big region, but where we have gene flow and where we have species interactions taking place is at the level of a subpopulation or a very unique population scale. And so a subpopulation is a local group of organisms of the same species. And we define subpopulations because they inhabit the same spatial location. Okay, they might not be inhabiting their whole geographic range, but a subpopulation inhabits one particular spatial location, one usually fraction of the whole geographic range for that species or population. Here we're looking at all the different little subpopulations of mountain sheep as they occur in the western United States. You can think of this in terms of these mountain sheep, and again, I think of them as like bighorn sheep, right? These are herds, and the herd is the subpopulation. The herd are usually all genetically related to each other in some form or fashion, and then they form metapopulations of interacting subpopulations when we have gene flow and interbreeding between nearby subpopulations. And so a metapopulation is one that is connected by gene flow, and there is movement of individuals between subpopulations within a metapopulation. And in general, metapopulations share a common climate. They share a common geographic range. all of the little herds of bighorn sheep in the western United States form a metapopulation that encompasses their geographic range here. They are a different metapopulation from the bighorn sheep that live in Asia. Because in Asia, there's no gene flow with the North American populations. And they might be the same species, but they're different metapopulations. And again, when we think about genetics and conservation biology, we have to manage for both of these scales. Okay, you have to manage what's happening at the subpopulation scale, and you have to be managing the metapopulation. So lots of higher level ecology today in landscape management focuses on connecting subpopulations into larger metapopulations. The idea of creating habitat corridors that allow subpopulations to begin to interbreed and intermix again to restore large metapopulations. This is the idea that, again, the whole western United States used to be covered by these enormous herds of bison. They were all interacting with each other because they were so big. They were all using their huge geographic range, and then they got hunted to near extinction. And there are a few remnant populations of bison on certain parklands or managed populations that are subpopulations of a bigger population of bison. If we really want the bison to be well managed, we've got to let our subpopulations interact with one another. We have to keep genetic variability high. Okay, cool. All right. We can then talk about species in kind of in two different ways. You can, as a species, you can have a ubiquitous population distribution. And ubiquitous, this means that you have a really widespread geographic distribution. You have big population sizes usually, and you have big subpopulations that really approach the overall landscape of a metapopulation, like just constantly by being there. And so the red maple and its distribution across North America, which is the kind of darker green area here, is the ubiquitous distribution. These tend to be generalist species that have broad habitat and resource requirements and that do well in a variety of conditions. They're intolerant, or they're very tolerant species. They can do well in a variety of soil types, a variety of precipitation and temperature regimes. They can eat, if you're a heterotroph, you can eat a variety of different food items. That gives you a ubiquitous distribution. You can encompass a large geographic range. Does anybody know what we would call it if you had a small geographic range? You were very specific in your requirements.
Specific distribution.
Yeah, but of course we have a special term for it, right?
Non-ubiquitous.
Non-ubiquitous, that's true.
I'm done.
You're an endemic
I know it.
species, okay? And again, it's like, if you
makes
know
sense.
it, it's great.
I knew it.
Like, what I tell you, you're like, oh yeah. Like,
An endemic species, that makes sense.
yeah, it does, because you're like, oh, I know what an endemic species is. It lives in one place. It's endemic, or very specific to a particular region, as a very endemic distribution. It's the opposite of ubiquitous, as a narrow geographic distribution. It has very specific habitat requirements. It's the panda that only eats bamboo and only lives in mature bamboo forests, okay? They tend to be specialist organisms like pandas that have really become great at doing one thing. And they are the best competitor in that environment where they find those resources, and they do crummy everywhere else, like pandas. We would all love to have pandas in North America, but we just never will. Okay. We would love to have polar bears in Antarctica, but they just don't live there yet. Okay. Our example here is this little blooming flower. It is a very endemic. It only lives on, it's the shale-bearing evening primrose. That's what it is called. It only lives on exposed hillsides with shale as the soil type. And that only occurs in very particular regions of the Appalachian Mountains. And so it has a very narrow geographic distribution. It will never grow in the Rocky Mountains because it has a different type of underlying parent material for the soil. It can only live here. It has evolved to live in this very particular place where it does great because other types of plants can't grow here as well. They're not as well adapted. But it also means it has evolved itself out of a wider geographic distribution. in general, ubiquitous species do better when we think about endangerment and extinction, because there will be remnant pockets of them in lots of different places. Endemic species have a high extinction risk because it's easy to accidentally bulldoze them all over. Okay, it's easy to accidentally build a neighborhood over their entire spawning ground because that's just how they're distributed. You have to know this about your population to be able to manage it. Okay, all right, so population size. We talked about population density and distribution. The next thing that we want to know about population is population size. And again, this plays into our calculations of population density, and it will affect distribution across the landscape. So population size is very simply the abundance of individuals in that population, the number of individuals. In order to determine population size or abundance, you've got to define the area that you're going to count as their area, okay? You have to define geographically what the bounds of your population are if you want to determine density, abundance, distribution pattern. Okay, the density then times if you have your density, so if you know there's always 10 individuals per unit area, you multiply it by the unit area, you'll get your total abundance. And so this is what ecologists do. They say, okay, we're going to go measure the density of individuals in certain areas and then multiply it by the total possible area, okay, in order to figure out the total population size. Why do they do it that way? Why don't you just go out and count everything?
Animals move.
Animals move. Plants don't. Why don't you just go count every plant in the forest and be like, hey, population size is this?
No, it's
Too many plants.
Exactly. You can't physically do it or you'll go crazy. Okay? And like honestly, this is how we use our time and our resources effectively, is that we take random samples and count population size and density there, then we expand it to include the whole population range to get an estimate of population size. So in this particular instance, kind of the same figure we've had at least once already today. They said, okay, we're going to count the population in these 3 little pockets. We take the average, we multiply it by the total area, we get total abundance. What I want you to start to recognize is that the way that individuals are distributed in the landscape, in this case they're quite clumped, will affect your estimate of population size. Okay? If every one that you counted was like 3, like say you counted 3 here, and then this one, and then this one, you get a really much lower population abundance estimate than if you counted 1 and this guy and that guy. Okay, you have to account for how your organism's life history and how you expect them to be distributed on the landscape when you go to assess density and thus do population size estimates. If you just go out there, you're likely to make a mistake unintentionally in population size estimates. And that can be really important. If you count a whole bunch of clumps and you're like, hey, there's 1,000 of these guys here, of course you can build a new neighborhood right next door. But there's not 1,000, there's only 500 because you counted where they were densest and they're clumped. That has a big impact. At the same time, if you went here and you counted the least dense areas and you were like, oh my God, I think they're almost extinct, but they're not, like that really causes you to spend money, taxpayer money, and your time as a research scientist quite different. Okay, all right, so ways that we estimate population size. And the first one, the thing that you guys have already been involved in, is what we call direct counts. And look, it's not you in the field, but it's someone that looks just like you. They have a quadrat, a meter squared, and they are counting individuals. And again, the idea is you take a sample area, you count all the individuals of the population, you multiply that by the total area, and you get the number of individuals in the whole area. Ways that work really well for this, again, are the quadrat idea, that you take an area of known size and you count all the individuals there. So in the case of the quadrats that we've been using, there are a meter on each side. You're counting a meter square worth of area each time. You then go, okay, there are so many square kilometers in the range. I estimate there are so many red maple trees in this given area. You can also do it based on a transect, very similar to what we did in the forest last Friday, where you take a transect of known length, usually it's 10 to 25 meters in length. You say, I'm counting the number of individuals in this area encompassed by my transect. You multiply that again by the total area, you get an idea of population size. Transect biology tends to work better for individuals that are more sparse and less dense across the landscape. If you just throw out a quadrat, you're not really likely to catch a raccoon or a large tree in your quadrat. If you do a transect, you're likely to encounter a raccoon or a raccoon burrow in a large tree. So again, your assessment method for drug counts needs to be sensitive to what you're actually trying to count. We've moved on in a lot of our ecology today to using drones and satellites, as we've talked about, in order to encompass bigger areas and to get better counts of individuals. And so quadrat and transect biology works great if you're working with plants or sessile animals, because they're not going to move very fast and they're going to stay put while you count. Okay. It works really poorly for birds, for fish that swim away, for big animals that are sensitive to hearing you coming out into the environment and will run away, and this bias your count. So drones and satellite technology work much better. Taking pictures or video from airplane as you fly over a region are much better for large, big-bodied organisms that move really quickly. For organisms like in the tropical rainforest that are very sparsely distributed, particularly for tree species in the tropical rainforest, you may find a large diversity of species in a given kilometer, but you may only find one of each tree species in a square kilometer of rainforest. And so you need to have a drone or a satellite take big pictures of the rainforest to actually get a population count for things even like trees. The other place, of course, where drones and satellites help us out a lot is terrain that it's hard for us as humans to actually get to. So we can't scale mountains and do transect biology. We can't go into conflict areas where there's actual war happening and do population assessments of things like mountain gorillas. Okay. We've actually been able to leverage technology to help us get better population size estimates of lots of different places. Okay. So again, the idea of the quadrat, you throw it out there, you have a known area, you multiply by total area. One of the key factors, again, to getting an accurate population size estimate is to make it random where you put your quadrat down. Otherwise, it's going to be biased by what's easiest for you to get to and easiest for you to count. It just happens. Or you can be on the other end of the spectrum and you go to the place where there's the most stuff and it's the hardest to count. To get an overestimate because of that. So what are some ways that you could get a random sampling if you're doing quadrat or transect biology? How could you make the choice of where you're going to count very random?
You have your cards, you have like random number generators.
Yeah, we use random number generator programs. And we tell the computer program, we say, hey, we're going out to this area, 10 by 10 square, pick me out 10 numbers randomly, row and column where I should go and put my quadrat. And in that way, it's chosen ahead of time. And you go out there, you look at your 10 options, and 2 of them might be too steep or covered in water that you can't actually count. And you get 8 really random observations of population density distribution. What I had you do, of course, was to draw random numbers along your transect or to choose a direction or a number of steps to go in an attempt to make it more random. Okay, it's influenced by what we can actually get to that day, could actually assess, but then I can try to introduce a bit of randomness for you to where you're actually going to make your observation. What is the disadvantage to doing quadrat or transect biology? Maybe we should start with what are some advantages to quadrat or transect biology?
It's fast.
It can be pretty fast, and especially if you're an expert in your field, you can go out to a quadrat and you can really quickly identify plants and count them. What else is good about it?
Cheap. I don't know. It's a little
Yeah, I mean, like you can go to
stick
Home Depot and make a transect for less than $5. And then as long as you can get yourself to the site, you can do a count. It is cheap.
I have some disadvantages now.
One other huge advantage is that you're actually counting real organisms. Okay, like you're there going, hey, that's really poison ivy. Okay. In satellite imagery, we have to make some assumptions about what we're seeing. We may not have the level of identification and ground truthing, we call it, that we do when we actually go out and do a direct count with a quadrat or on a transect. What are some disadvantages?
Could at the same time be time consuming because you might have to, you can't go in the winter. So if you decide to do it during the winter, you're gonna have to wait until it's spring or until it's summer to where you can actually go out there and get a good sample because there's not snow on the ground.
Right, yeah.
And
So
it could also be dangerous.
It can be dangerous. It can be very time consuming to identify and count every plant. I have a friend that is like, this is what she does, is she goes out and does quadrat counts of different native plants in different areas on Vancouver Island to get population size assessments to manage Vancouver Island as an ecosystem. And like, they'll do like 10 quadrats a day when they're in sampling season. I mean, it's not fast. And it takes like 4 people to assess a quadrat because you got to have your fern expert and your wildflower expert and your lichen expert and your moss expert all standing there at the same quadrat area doing their thing. And so that's time consuming in terms of actual physical time. Imagine if you have 4 PhDs standing around a meter square counting stuff. That's expensive when it comes to like salary and like expertise. So sometimes it can seem quite wasteful. But if you want to ground truth stuff, you want a real count, you gotta go and do it. The danger part of it is very real, in that you can be in an environment that is hazardous to your health. There could be pollution there could be wild animals there. Even in plant biology, you can run into nasty stuff. Again, I had a friend that was doing quadrat biology in Australia, counting plants, and she ran into one of these stinging nettle vines, and she had these big blisters all over her face for a couple weeks. And like, if you're out in the bush country of Australia with a painful, weepy rash, like that's not fun. Like no one is taking you to the doctor because you're supposed to be out there for 3 weeks. Okay. So there's these things. Okay. Like it's cool. It's fun to go out and see the stuff, to identify it. Maybe you'll discover a new species. There's downsides.
Maybe you'll get in the face with stinging net.
Yeah, maybe. Oh God, she also talks about being chased by angry male cassowaries. These big land-based birds, kind of like ostriches, that live in Australia and New Zealand. And the males fight each other for territory. They have these big claws on their legs that can actually slice through truck tires. And they have these big domes on their heads that they ram into each other with, right? And if you get a male cassowary that has been in a pocket of territory where it hasn't encountered another cassowary for a long time, they tend to be really over-aggressive. And if you walk into their territory, even as a human, they will attack you. Wasn't expecting to be chased by a cassowary.
Okay. The scary birds.
All right. The other thing that I want to talk to you guys about today in estimating population size is what Indy brought up earlier, is this idea of tagging individuals in a process that we call market capture. And so this weekend, actually, we were tagging monarch butterflies with the zoo at one of their prairie restoration sites out near where I live in Mason. And we're sticking these little plastic tags on the butterflies here, and noting that we caught them here, in hopes that they will be seen in Mexico, where they are migrating to by the end of November. And that the scientists in Mexico then will count the number of tags and get an idea of population size. They will also, in recovering that tagged individual, get an idea of migratory routes, what habitat was used along the way. That tag is seen in a couple different places on its way to Mexico. Okay, the way that this works is that first you have to go out into the environment and you've got to capture and mark some individuals. And so the way that the math works here is that the ratio of originally marked individuals to total population size okay, you have gone in and you have marked a portion of the total population, okay? But you don't yet know what that ratio is. You just know how many tags individuals you have, okay? Tag 10 butterflies. I think this weekend we totally, as a group, we tagged like 28 monarch butterflies. We caught a ton of monarch males, so we weren't really great at catching monarchs. Okay. The next thing that you have to do is you have to go back to that same place, okay? Or another site along the, geographic range of these organisms, and you've got to recapture organisms again. But the second time you go, you're not necessarily gonna tag. You can do some tagging too, but that kind of goes into your future estimates of population. You're gonna go back to the site, and you're gonna capture organisms again. And you're gonna record the number of tags you got. The number of tags that you recovered versus the total number that you caught that day. And that proportion of tags to total population that you caught on day 2 should be the same proportion as the tags you put out there on the first day to total populations. And that is how this math works. So we want to know total population size. And we're going to know the number of tags we put out initially. We're going to know the number of tags we recovered when we looked again. And we're going to know the total number that we captured when we went to look again.
So is total captured on day 2 just the ones you captured day 2, or is it the
It is
day 1 and
just,
day 2?
no, it's
Okay.
just the ones you captured day 2, but it includes both marked and unmarked individuals. Okay.
Oh, so
So this is equal to marked plus unmarked
okay.
or untagged, but only on day 2. Okay. Okay, cool. All right. So marked to your population is equal to the number of tags recovered to the total number of captured. And again, you can do this in lots of different ways. It doesn't have to just be you that goes out. It can be fishermen or hunters that recover tags. And they say, oh, okay, when we brought in our load of anchovy today, we had 10 tags out of 1,000 anchovy that we caught. Okay, and that gives you the ability to start working with this type of map to get an idea of total population size. Okay. Oh, right. Okay, so we're going to try this mark and recapture method for estimating populations. Okay. So I have a worksheet for you, and I've brought these things with me on several occasions. It's for you and a partner. You can choose your partner. If you want to work alone, you definitely can work alone. You need a fish map, and there's a few different types of fish, and you'll work through all of them. There are 3 types of fish. You need 2 dice, and you need a worksheet. And I will let you try it.
I don't lie. I'm tired. You don't work. I just woke up. I don't tired. Yeah, that's because I haven't had
Okay.
breakfast.
All right. When you have your partner, come get a fish mat and some dice, and I'll give you a worksheet.
Yeah, too far. It's not far. It's too lazy to go here.
You can choose where you want to start. You'll do all 3 fish species. There's salmon, there's herring, and I think I have.
You want to
You'll
the
do all
herring or
3.
the team herring for sure.
Okay.
Can we just put the our names on?
Oh, yes, yeah. One worksheet, so just put your name on it. That's how you get participation.
Sorry.
She?
All right. Don't worry about it. These fish things are surprisingly old and have held up well. So there's like 2 sheets in the rest. One on the other. This whole thing. It'll tell you how to do it, okay? The idea is you roll the dice, you open up a flap,
You're
Just
gonna
fine
roll
by
the
the
dice,
dice.
okay? Alright.
and you record how many marked and unmarked and total fish that you have. And the red fish are tagged. And then you write, okay, I got one red fish out of a total of 6 fish. Okay. Okay, do it.
Roll dice, open the flap with the same number.
7. Alright. 1, 2, 3, 4. 1 out of
Where did they
7.
go?
No, you don't need.
1 red fish out of 7.
I have 6 dice.
So,
Oh, no, they're hiding. I was like, no. That's not where I would find that.
1 red, and there's 6 blue fish. So it's total 7 fish.
It's just blue and red right now, and you'll total in a second.
Why does it look purple to me?
Because there is, I'll tell you in a minute. There's 2 different color blues, but they're all blue.
Okay.
Okay.
Alright. So you want to roll? Or-
Sure. Here, I rolled 7.
Same one.
Oops.
If you go to the same one, go to the same one, right?
Yay,
That's
look at
the, telling
that.
you, hey, you gotta be
Yeah.
random about it. So if you go back to the same square, go back to the same.
Alrighty, I got 11.
One, or, what?
2 red fish and 1, 2, 3, 4. 5.
You wanna give it a go?
2 red.
1, 2, 3, 4, 5. Okay.
You got 9. 1 blue fish.
Oh, no.
I'm just expert at this. Expert at getting this.
I also got 9.
Let's do that. We're done.
Alright, so now it's the next. Sum of all numbers.
19.
6, 10, 17.
3. Oh, we got 4, sorry. I was like, 3, no.
Hold on. What is 19 plus 4?
23.
Classic car.
Number of initially tagged equals 10.
So I'm telling you then that I marked 10 fish. There were 10 red fish total out there to be caught. And so you can calculate total population size by taking the total number of fish caught times 10 and dividing it by your number of red fish. Okay? And again, you're gonna take, remember when we have proportions, you can cross multiply. Okay?
Oh,
So you're
yeah.
gonna cross multiply the total number you captured times 10, the number I'm telling you on mark. And then you divide it by the number of red fish, the number of tagged fish. That'll give you a population size. See the calculator.
So
10.
our tag for day 1 would be 10, but
Yes, I'm telling you that I put 10 red fish out there for you to go back and count.
Right.
So,
I'm just gonna look at the fish for a second.
so it's 23 times 10.
the total pop,
230. And
That's
divided
what we
by.
want to know.
I don't know, it's like, yeah, this is a
the
P
total
down here.
captured is,
So all you have to do is this. Total number of fish caught
Yeah, it's so hard to do that.
times 10, 22, divided by 5.
on the tag.
I
57 times 5.
Alright.
I'm gonna get a
That
snack.
is the total
Okay.
fish.
And it's right, you can use it or not.
Alright.
There you go.
I think it's good.
Total population?
Or that's,
44.
I
Okay,
think it's good.
get another type of fish. So note that you just did salmon.
We did a yellow tag fish.
Yellow 10. Weird name.
Yellow tag.
Isn't it tiny or something?
It's so weird.
Red herrings.
Oh, yeah.
What we got now?
Something called migrating herring.
We got herring.
One.
Alright.
You just there.
We got herrings.
And,
Alright, do you want to roll first or you want me to?
Doesn't really matter. 'cause it's
Oh,
coming.
you
We're
roll.
taking turns.
They're like red herring.
4. Alright, let's look at that. We got 2 red.
Oh, 2 red, great.
Yes.
You never watched them do?
3 red.
And 1, 2, 3, 4, 5, 6 blue.
Oh my god, it was serious.
Cool, now your turn.
And you three went out little.
4. Same information.
Oh.
It's a 5, by the way.
Where did it go?
It's in front of you.
I guess.
Alright.
My turn?
Yep. Alright, so 9, 10.
We got two for.
Oh, you can flip it
Zero.
over on the back if you want to use the different.
Oh.
There is a back that you can like do the different.
That is not.
That makes it easier.
3-1, okay.
Five.
Just gonna mention that 10's a nothing.
One in each.
We got a total of nothing.
Interesting.
Big old
Yep.
pile of fish.
Oh,
Wait,
look at that.
migrating.
A whole no fish.
Okay, where do, where
Yeah,
are we?
0, 0.
Alright, 0, 0. We got a whole nothing. Alright, so it's my turn now.
32.
One with red.
What do you call that?
It's up to you if you want to count it.
I can't
Oh,
tell.
I'd
if
say
the-
2. Yeah, it looks to see like
Because-
it's in the line,
Yeah,
and the top has gotten a little bit misaligned. I would say 2 blue and 1 red.
I was about to go like, do we count this 2.5? I mean, 1.5 fish.
And
Alright.
so usually when you do quadrat biology, you make a rule for your group ahead of time that says if any part of its body is outside the quadrat, we aren't counting it. Or if any part of its body is in the quadrat, we are counting it.
One.
Alright.
You have to set a rule so that you don't decide in the field.
Ah, same thing.
Same information. Woo.
Count one.
12, second position. 16, 4, 6. What is 16 for 1?
16 plus 6.
So,
Alright.
22, yes.
you can use stomach contents to do population size estimates.
I wasn't being serious, man.
My
No,
turn.
you can't.
It's okay.
So especially in like fish species where it's hard to get out in the environment, like think about how much more difficult
That's
it
a
is
good
to do
one, Randy.
transect in the wow.
Oh, yeah.
You have to
We got
get
one.
in scuba and snorkel and do it, right?
Alright.
I get it.
Now we get another thing that you're gonna
So,
No, I like you.
Yeah.
Fishing, yeah, but
Our last
then
fish
you
you
go
can
Yeah.
map.
so we do use that. The other thing is we use their stomach content.
5 again.
They eat a lot, those little things, there's a lot of them. That's why.
Oh,
Okay.
That's weird.
Alright.
When you
Salmon.
have your population estimates for all 3 species, come write them on the board in categories. They belong in.
okay. Give me that.
4.
It's not
We
bad.
got 3 blue
Well worth the
fishies
money.
and a red fishy.
Ooh.
So 260.
Alright, we got 9. 3 blue fish.
Oh, that's my favorite. That's my.
Alright, we got a 10,
2, 3. That's my
1, 2,
sum of
3,
all my
4
red.
blue fish and a red.
Why did I say 5? Okay, so it's. Yeah, red fish. So it's 3.
10.
I just said 5 since we got.
Same thing.
4 blue.
Okay.
Okay.
Oh, my bad.
7.
11. It was 11. Which I accidentally missed.
Wow, no, we kind of heard this all the
4
time.
blue fish and a.
It's like the same thing.
It's weird. And we went to a different one.
10.
4 blue fish and a red. We got to the same thing 3 times.
We're gonna talk about that.
44, 64.
Also, I'm just gonna say we could
Which
never
one is
get,
which?
we technically could never get one
Salmon 44.
So salmon 22.
unless we threw away a die. We could never get one.
Yeah, salmon.
I think we did more than
Wait,
one.
I looked.
He was so confused.
No,
I'm so sorry, Olivia. I looked
No, I
on
think we
this
did
line.
Instead of
more
looking
than-
down here where we actually.
like we're supposed to.
Oh, we did one more than we are supposed to.
God, you know, like that is not one of the numbers that you just said. Okay, sorry. 64 is hang. Yeah. and then 87 is herring.
All right, once you have your numbers, go add them to the board.
You're right there.
Hey, can you do that?
Not really, but close it, but
I guess so.
I can do it. I don't think.
Nice move, Jimmy.
I like it that it's with a K, Carl. Carl with a K.
Hey, do you want the paper?
Sure.
What do you do? If we have a half hour, we just round that then.
And again, we're have this a little bit of this at the beginning. When you're doing quadrat or transect biology, you have to set rules. If they're, if it's half in, are we gonna count it or not? If it's a half number, are we gonna round up or down? And there are rules for significant figures and rounding, and if you play very strictly by them, then you don't have these problems. Okay. All right, as we are looking across our different fish species, did we get the same number for all of them?
Not at all.
Guess what? On every single one of these fish maps, there were 40 total fish.
Ooh.
The population size was 40 for all of them. Okay, so why do we get these different estimates when there were 40 fish on every map?
Because for whichever one that we lifted up, there could have been more than, or we lifted up the same area a couple of times.
Good. If there's bias in where you're looking, you might bias your overall estimate. And so when you're rolling the dice, like you can never roll a 1. Okay, there's one area you're never gonna get to. It's really likely you're gonna roll a 6 or an 8, right? Because that just happens more frequently in rolling 2 dice together, okay? So if you're biased about your observations, even though you're trying to be random about it, it'll affect your estimate. What else can affect your estimate? Population size.
The number of samples.
Good. You only did 3.
Yeah.
And assuming that you did this and you only did 5 little windows at a time, if you increase your sample size, you should get closer to the true value. You should improve your accuracy.
How many samples are good?
So how do you know? You have to do some modeling ahead of time to know how many individuals you need to count or how many species you need to count. Or this idea that you keep adding observations and then and you're changing number, changing your average, and you keep adding observations and your number keeps changing. And you know you're at that saturation. When new observations quit changing the answer, then you can stop. What else would affect that we've talked about today? What else that we've talked about today would affect your population size? Oh yeah, if you have a higher proportion of tags, you'll tend to get a better estimate. What else?
How you drew them on there.
How they're distributed across their environment varies. How the estimate goes. Okay, so the tag are clumped around their reef habitat. So if you measure a clump, you're going to increase your estimate of population size beyond 40. And it's not uncommon if we had a whole bunch of different groups that you have some that were really high, you have some that were really low off the scale because they just managed. Our herring, again, is either low or it's high. Because our migrating herring, again, are clumped into schools. Okay, they're moving together across the landscape. And so it might look random or uniform, but they're actually schooling. And so again, they end up with this kind of different distribution. And that will affect the population size. The salmon did pretty good. There was actually one, that was like 44. The salmon have a much more random or uniform distribution when they're just out in the ocean. When they're in their little streams, it's pretty bad. When they're out in the ocean, though, they show a pretty random, pretty uniform distribution. So the population size estimates tend to be a little more hard. And the reason there are 2 different blues is because when I originally made the maps, there were 30 fish, and the estimates never came out right when students did, so I added 10 more fish, blue fish, with a different blue. Fish. Okay, cool. There are a whole bunch of assumptions that go with using the mark and recapture method, and I will place them on a slide and we'll talk about them briefly when we come back to class on Tuesday. But the final thing that I want to leave you with is that you can measure population size. Oh, wait, come on, guys. So population sizes change over time, which is what we want to talk about next. Cool. All right. I'm going to hand you homework 3, which is due October 15th. As you